All right, so I want to do a review with you guys for the quiz that's coming up over DNA and RNA, and then also cellular movement. Like the sign says here, it is Wednesday of this week, so right now it's Monday at about 11 o'clock. I'm trying to go ahead and get this put up to where you can have 2 days to watch it. So we'll just go through the quiz like we usually do. The process that moves water molecules across the membrane of a cell is osmosis. Again, osmosis, remember, is a passive transport, meaning it does not need any extra energy. And when we talk about osmosis, it is the specific movement of water molecules across the cell membrane or plasma membrane. And what they're trying to do is they're trying to move to create that dynamic equilibrium. Taking the process of taking material into the cell where the cell membrane enfolds on itself or comes in on itself is endocytosis. So bringing substances into the cell by the enfolding of the cell membrane is endocytosis. It is a form of active transport, meaning that it needs energy. Chemical changes in the structure of a chromosome can happen based off exposure to some mutagen agents, exposure such as like formaldehyde, any kind of X-ray or radioactive material. Sunlight can even do that to a gene. Basically, what happens is you get a mutation, meaning that the DNA strand is changed in a different way. Now, remember, when we go from a line of DNA that's inside the cell, okay, remember it's a double helix, and when it splits and we make a copy of itself, that's called DNA replication. But when we make a line of mRNA, we call that transcription. That line of mRNA is going to go to the ribosomes where the tRNA brings in an amino acid connected to it, and the codon, the 3-base pair, matches up with the anticodon, which is also a 3-base pair that's connected to the tRNA. It also has an amino acid, and that long chain of amino acids being put together is what we call a polypeptide bond, which is multiple amino acids coming together, or what we call a protein. We know that because ribosomes make proteins when we studied the cell organelle parts. So that's all putting it all together. The blueprint for a protein is originally carried by the DNA molecule. Okay, it is the genetic code, and one gene codes for a protein. Translation of the DNA sequence, we have what we look at is we look at the sequence of the 3 amino acids linked to the peptide bonds. So the anticodon or the tRNA is where translation happens at the ribosome. So we're going to look at that 3 amino acid linked to the polypeptide bond. Just like I said before, when we talk about transcription, that happens right before translation. Translation, again, happens at the ribosome. So we got to have that mRNA line come from the transcription, DNA transcription, which happens in the nucleus. And it precedes protein synthesis or translation at the ribosome. You're going to have to remember some different things when we talk about hypertonic, hypotonic, isotonic solution. Remember, hypertonic solutions pull water out of the cell because the water follows its concentration from a high to low. So if we give you an example of like pickles in a preserved brine solution that contains, a lot of salt and some other ingredients, the cucumbers are going to shrink while they're in this pickle juice, the pickling agent, because the salt content's higher in the solution. So that would be a hypertonic solution. The correct sequence of molecules as a gene goes from coded information in the nucleus, meaning DNA, excuse me, to the product the cell wants to make, which is a protein. It goes like this. It goes DNA, messenger RNA is made off of the DNA, and then the transfer RNA brings the amino acids to the ribosome, and we have a long chain of those amino acids put together in a protein. What's the accurate description of the process used to turn the DNA code of a gene into a protein? Transfer RNA will deliver specific amino acids to the ribosome, matching the messenger RNA codon. So the anticodon from the transfer RNA is very specific, and it carries an amino acid to the messenger RNA's codon, matching up, and that's how we get the protein made. You'll have to be able to look at the codon charts, understanding that the left rows are the first base in the anticodon. The top columns are the second base, and then the third base would be broken down on the right side. So you're going to have to be able to read those. Nutrients enter a cell with the concentration gradient by the process of diffusion. Diffusion is a movement of nutrients from an area of high concentration to low concentration. And what they're trying to do is they're trying to achieve equilibrium. Again, the process of water crossing a selectively permeable membrane is called osmosis. Anytime we talk about water movement, that is osmosis. A gene is a segment of a chromosome that codes for a specific protein. So as you look at the chromosome, a certain section of that is the gene, also known as a sequence of nucleotides found in the DNA of this chromosome codes for a specific protein or trait. The segment of DNA is known as a gene. That will also code for traits. Genes code for traits and can be located in the DNA. So, for instance, if I talked about osmosis again, if I was to put like a raw shellless egg was placed in a sugar solution, the egg shrank. The egg was brought back to its original size by placing it in water. Again, we're talking about movement of water in and out of cells, so that would be osmosis. Plant cell remain unchanged in size or shape when I put it in a solution. We would talk about the solution being an isotonic solution. The effect of placing a cell in a hypotonic solution, the cell would expand, remember, because the concentration would be higher on the inside of the cell. So that means the cell would expand and become turgid, or it would stand upright, because, again, we're trying to get that dynamic equilibrium there. So what we would have is we would have the cell expand because the water would move from the solution, which is under in the salt concentration, and go into the cell, which would be higher. The central dogma of molecular biology is centered upon the process of protein synthesis. We are making proteins in which the information from the DNA is transcribed first in the nucleus. So an mRNA line is transcribed off a DNA line, and then that mRNA line goes to the ribosomes where it's translated, resulting in amino acids being joined together into polypeptides or proteins. The central dogma of molecular biology is centered upon the process of protein synthesis, in which the information from the transcribed and translated, resulting multiple amino acids being joined together to form proteins. So we call it protein synthesis. The actual thing that we're trying to get is proteins, which help us to, excuse me again, help us to carry out functions within our cell. When DNA replication occurs before meiosis, the original DNA strand could be a number of bases, could be 4 groups, 5 groups, sets of codons is copied into complementary strand. And if we lose a set of those those codons, we would call that mutation a deletion mutation. On the opposite hand, during DNA replication, when it occurs before meiosis, if we have an extra set of codons, that would be what we call insertion. So insertion would be adding more codons or more bases after meiosis or before meiosis during DNA replication. And if we lose some, that would be called deletion. During diffusion, molecules move from a high concentration to a low concentration. When you look at flow and net flow of of particles in a solution or in anything that we talk about diffusion or osmosis, molecules are moving from a higher concentration to a lower concentration because they're not needing any extra energy. Um, if carbon dioxide molecules are shown moving from one side of a medium to another, which is the best describes the reverse of this process. So if I have them moving from a high to low, but I need them to move from that low to high, we would be talking about active transport. And remember, active transport requires energy to move that against the concentration gradient. DNA is a double-stranded helix, much as when we compare it to RNA being a single, and can be described as ladder rungs. The rungs are actually the nucleotides. The DNA are the building blocks of DNA. A sequence of 3 of these nucleotides is known as a codon. Codons are important in the composition of proteins because each codon calls for a specific amino acid. Okay, remember the codon chart there is going to bring the amino acids. So DNA encodes the information necessary to produce the proteins needed by your body to make the proteins. DNA first undergoes a process known as transcription. This is when the information on a DNA strand is transferred to a molecule similar to the DNA known as RNA. So as we move from the DNA to the RNA, messenger RNA, we are actually moving information to be creating or to be used to create proteins. When we look at the central dogma, the DNA codes for the production of mRNA during transcription. In eukaryotic cells, the mRNA is processed and migrates from the nucleus to the cytoplasm. The messenger RNA carries coded information to the ribosomes, where this information is translated and used for protein synthesis. So the 3 areas that are incorporated into the central dogma is the nucleus, then the cytoplasm, then the ribosome. When we look at cell drinking and cell eating, they're both forms of endocytosis because we are bringing things in. If we were going to get rid of something, it would be called exocytosis. So hopefully you spend some time studying and getting ready for this quiz. If you have any questions, make sure you check with me today or tomorrow in class.